Okay, today we are talking about cellular respiration, Chapter 7. Cellular respiration, the equation is kind of the opposite of photosynthesis that we talked about last time, but the process is an opposite exactly. So we're going to talk about respiration. We're going to review some terminology. Then we're going to break it down into aerobic respiration versus anaerobic. We're going to talk about how things are synthesized and broken down, and exercise. So, cellular respiration is basically the conversion of carbohydrates into ATP, which, remember, ATP is the energy for the cell. So basically, we are taking the carbohydrates that were processed and made in photosynthesis and now using them to generate ATP for our cells to do work. So ATP, again, provides just the right amount of energy for the cell to do its work without any waste. And glucose is the most common molecule that we use to generate ATP. Conversion of glucose to ATP is about 39% efficient. So while that doesn't sound very good, it actually is pretty good because we lose a lot of energy as heat. So most of the energy when we transform glucose to ATP is actually lost as heat, also called metabolic heat. So glucose is a type of carbohydrate, and remember, it's a simple carbohydrate. It is the building block, basically, for a lot of other carbohydrates. And why is ATP used for cellular work and not glucose? Glucose is used for a lot of other things. Why don't we use it for cellular work? Because ATP releases just the right amount of energy, and glucose actually has more energy. So if we use glucose to do our cellular work, we would actually be wasteful because so much energy would be released from glucose at once. It would be too much for what we are doing, what our cells are doing. So in order for our cellular processes to be efficient and effective, we use ATP instead of glucose so that we don't waste a lot of excess energy. So here is the equation for respiration. You have a carbohydrate plus oxygen gives you carbon dioxide, water plus energy, so ATP. Remember, oxidation means that a molecule has lost electrons or hydrogens, and reduction means that a molecule has gained electrons or hydrogens. So remember, the electron has a negative charge, so if you gain it, it's kind of reduced. Remember that? So when you look at the equation, you have a carbohydrate that is going into a carbon dioxide. So that it becomes oxidized because you can look at the hydrogen, and the hydrogen from the carbohydrate is now gone in carbon dioxide. So it has lost hydrogen atoms. So when you ask which reactant is being oxidized during respiration, it's the carbohydrate. Now, if you look at the oxygen is actually being reduced to water because it's gaining hydrogens. So if you're asked what reactant is being reduced during cellular respiration, it is the oxygen. The oxygen is reduced to water, and then energy, which is ATP, is created in the process. So glucose, C6H12O6, going into carbon dioxide and water, is an exergonic reaction. It releases energy. You're breaking down glucose into other products. On the other hand, ADP plus the phosphate to synthesize ATP is endergonic, meaning that it requires an input of energy. So we can have an exergonic fuel the endergonic, and it's called coupling, remember. So breaking down that glucose molecule in an exergonic reaction can fuel the creation of ATP. So the energy release drives the other reaction, as I said. So these reactions are coupled. Okay, so some terminology. Aerobic means with oxygen. Anaerobic means without oxygen. So does the chemical equation following reaction represent aerobic or anaerobic? All you need to do is look to see if there's oxygen present. We can see there's oxygen present, so that means it's aerobic. You have glucose plus oxygen yields carbon dioxide, water plus ATP. So that's aerobic. The next equation, you have glucose yields another carbohydrate, carbon dioxide, and 2 ATPs. So that's anaerobic. So you have C2H5OH, which we're going to talk about that later on. This is an anaerobic process. So aerobic with oxygen, anaerobic without oxygen. So you have more energy generated with aerobic than you do with anaerobic. And again, we're going to talk about this in a few minutes. So this alcohol, this C2H5OH, is actually a high energy product. Carbon dioxide and water are low energy products. But again, more energy, ATP, is generated with oxygen than without. So oxygen has to be present in order for glucose to be completely oxidized to carbon dioxide. So we're going to talk about these two processes. We have aerobic cellular respiration, generating 36 ATPs, and we have anaerobic fermentation, which generates 2 ATPs. There's a big difference in these processes when you're talking about with oxygen or without. So, substrate-level phosphorylation. This is the first part of cellular respiration. Basically, we are going to generate ATP. The phosphate group is going to be removed from the substrate and added to ADP. So substrate-level phosphorylation occurs during the first two processes of cellular respiration, glycolysis and the Krebs cycle. However, it only generates 4 ATPs, so it's not very efficient. So we have cellular respiration is broken down into 3 steps: glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis is the first step, and glycolysis does not need oxygen. However, the Krebs cycle and the electron transport chain do need oxygen. So if we go through glycolysis and oxygen is still not present, that is when we go to anaerobic fermentation. Okay? So oxidative phosphorylation happens in the cristae of the mitochondria. This is the last step of the cellular respiration, so the electron transport chain that I was talking about. So it's driven by hydrogen atoms being dropped off at the electron transport chain by the electron carriers NADH and FADH2. Those come from glycolysis, the preparatory stage, and the Krebs cycle. This includes the electron transport chain being coupled with chemiosmosis to generate 32 to 34 ATPs. So we're in the mitochondria now, in the cristae, and this is the final step of cellular respiration. So again, NADH and FADH2 come from the first steps of cellular respiration. They bring the electrons, they donate the protons, and pump them into the inner membrane space. That's going to fuel the ATP synthase pump to generate ATP. So, you have glycolysis. This is the very first step of cellular respiration. It occurs in the cytoplasm. What happens is glucose is broken down into 2 pyruvate molecules, which are 3 carbon molecules. So a 6-carbon glucose molecule is broken down into 2 3-carbon pyruvate molecules. Again, it occurs in the cytoplasm, but it only nets 2 ATP. This is substrate-level phosphorylation. It also nets 2 NADH. So just like we had NADPH in photosynthesis, now we have NADH. It's an electron carrier. So it carries electrons and hydrogens. And as you can see in the picture here, it's going to carry them all the way over to the electron transport chain. After those 2 pyruvate molecules are created, they will go into the mitochondria for what's called the preparatory stage. This happens in the mitochondrial matrix. The preparatory stage is when the pyruvate molecules are chopped off into acetyl molecules and they bind with what's called coenzyme A. So we get 2-acetyl coenzyme A produced, and carbon dioxide is actually given off as a byproduct from that carbon being chopped off to form the acetyl groups. So the acetyl coenzyme A will enter the Krebs cycle, which is the second official stage of cellular respiration. Through the Krebs cycle, again, it's substrate-level phosphorylation, and we only generate 2 ATPs. 6 NADH and 2 FADH2, however, are generated, so they carry those electrons and those hydrogens to the electron transport chain. FADH2 is just an electron carrier, just like NADH. So the electron transport chain happens in the cristae of the mitochondria, so those inner foldings. This is the most net we have. 32 to 34 ATPs are netted, and it's through oxidative phosphorylation. So basically what happens is we have all of these protein complexes, and the electrons go from complex to complex to complex to complex. As they're going from complex to complex, the hydrogens are being pumped through the ATP synthase pump. So they're creating ATP. So this is how all of that ATP is generated. Now, oxygen has to be present because as soon as we finish glycolysis, now we need oxygen. So if oxygen is present, we will go through the Krebs cycle and go through the electron transport chain. Oxygen is the final electron acceptor. So without oxygen, the whole system will break down. So the electron transport chain needs that oxygen to generate the most ATP. So as I just said, the electron transport chain through oxidative phosphorylation generates the most ATP. So during aerobic cellular respiration, 36 to 38 ATPs can be generated from one molecule of glucose. So if we go through the whole process, 36 to 38 ATPs will be generated from that one initial glucose molecule. So if oxygen is present, glycolysis will be followed by the preparatory stage, the Krebs cycle, and the electron transport chain. Again, oxygen has to be present for these reactions to occur. So glycolysis, you have the glucose broken down into 2 pyruvate molecules, 2 NADH being transported to the electron transport chain, 2 ATP being generated. The preparatory stage, you have those pyruvates being cleaved into acetyl groups, and they bind with coenzyme A to form acetyl coenzyme A. That then goes into the citric acid cycle or the Krebs cycle. It's the same thing. And again, we'll generate 2 ATPs, 6 NADH, and 2 FADH2s. Those will carry the electrons and those hydrogens to the electron transport chain, where most of the ATP is going to be generated. So C6H12O6 is the reactant, glucose. The product is going to be carbon dioxide because carbon dioxide, as I said earlier, is given off when the acetyl groups are formed from the pyruvate groups. So hydrogen atoms are being slowly stripped off of these glucose and kind of shuttled to the electron transport chain by NADH and FADH2. So in the presence of oxygen, one molecule of glucose can yield 36 to 38 molecules of ATP. 4 are generated via substrate-level phosphorylation, so glycolysis in the Krebs cycle. 32 to 34 are through oxidative phosphorylation, so the electron transport chain. The hydrogen atoms are supplied by the glucose, and they're carried to the electron transport chain by mitochondrial enzymes, by coenzymes. So NAD plus carries 1 hydrogen, forming NADH, and FAD carries 2 hydrogen, forming FADH2. So NADH and FADH2 are the electron carriers. They also gain hydrogen. So when they pick up hydrogens, they're gaining electrons and hydrogens, so that means they're being reduced. So the electron carriers are being reduced. So overall, glycolysis, your inputs and outputs. Your input is glucose. Your output is pyruvate. Again, it happens in the cytoplasm. It doesn't need oxygen, and it nets 2 ATP. The preparatory stage, pyruvate is what goes in. It binds to coenzyme A after it's cleaved into acetyl groups. So acetyl coenzyme A is the output. Happens in the mitochondrial matrix. Yes, it needs oxygen and does not generate any ATP. For the Krebs cycle, that acetyl coenzyme A goes in and binds to the oxaloacetate. So that's the input. Output is citrate. The citrate will go back to rejuvenate the oxaloacetate. The Krebs cycle, notice, is a circle, like the dark reactions in photosynthesis was a circle. So it's a cycle that keeps going and regenerating itself. This happens in the mitochondrial matrix, requires oxygen, 2 ATP are generated. The electron transport chain, we have hydrogens going in, oxygens going in, water is the output. Happens in the mitochondrial cristae, oxygen's required and used, and we generate 32 to 34 ATP and oxidative phosphorylation. So aerobic cellular respiration includes glycolysis, which is an anaerobic process. So if you talk about aerobic cellular respiration in general, you're talking about glycolysis, the Krebs cycle, and the electron transport chain. If you're specifically just talking about anaerobic respiration, then you're talking about glycolysis and fermentation. But in general, glycolysis is always going to happen. So that's the first step of aerobic respiration, even though it's anaerobic. So here are all of the nasty steps for glycolysis. You guys do not need to know all of those, but glucose is what goes in, pyruvate comes out, 2 ATP are required, 2 ATP are also netted, and 2 NADH are netted. So your input, glucose and 2 ATP, your output, 2 pyruvate molecules, 2 net ATP, and 2 NADH. So when oxygen is present, the pyruvate is going to bind with coenzyme A. It's going to be cleaved into acetyl groups, and carbon dioxide is going to be a byproduct. So the input is pyruvate, the output is acetyl coenzyme A, carbon dioxide, and NADH. There's 2 of those molecules. The Krebs cycle turns twice for every glucose molecule. And again, it's a circle, so it's kind of self-circulating. So those acetyl groups go in with the oxaloacetate. Once again, carbon dioxide is given off as a byproduct. 2 ATP are generated. 6 NADH and 2 FADH2 are also generated. So you do not need to know the steps, but you need to know the inputs and the outputs. So acetyl goes in, NADH, FADH2, ATP, and carbon dioxide come out. The electron transport chain is the big moneymaker, so to speak. So NADH go in, FADH2 go in. These supply the hydrogens and the electrons for the electron transport chain. If you look at the picture there, you can see that there's protein complexes that the electrons flow from complex to complex to complex. It's like a slinky going downstairs. When a slinky goes down the stairs, it kind of goes from stair to stair to stair. That's what's happening with these electrons. They're going from complex to complex to complex. The hydrogens being released fuel the ATP synthase complex that generates ATP. So as the hydrogens flow through the ATP synthase complex, ATP is created and generated. So inputs NADH, FADH2, and oxygen, because that's the final electron acceptor, outputs 32 to 34 ATP and water. So the electron transport chain is really where most of the ATP is generated. So here's a picture of mitochondria. You have glycolysis, glucose to pyruvate, the prep reaction. Again, the pyruvate is cleaved into acetyl groups, binds with coenzyme A. Acetyl coenzyme A then goes into the Krebs cycle. 2 ATP are generated there. NADH and FADH2 carry electrons and hydrogens to the electron transport chain, where 32 to 34 ATP are generated. Oxygen is required. It's the final electron acceptor. So here's an overall summary showing you the start of glucose in the cytoplasm. It goes through glycolysis, 2 ATP, 2 NADH. Pyruvate is going to be cleaved into acetyl groups, bind with coenzyme A, give carbon dioxide off as a product, create 2 NADH. Krebs cycle again creates 2 ATP as a product, gives off carbon dioxide as a byproduct, 6 NADH and 2 FADH2. Now all of those NADH and FADH2 carry the electrons and hydrogens to the electron transport chain, where 32 to 34 ATPs are going to be generated. So some interesting facts about ATP. We have enough ATP to sustain life for about a minute. Some poisons inhibit ATP synthase, so you cannot make ATP. So cyanide, for example, inhibits ATP synthase, so you can't generate any ATP. If you can't generate any ATP, you do not have energy for your cells to do anything. Some tissues have more mitochondria than others. In chickens, for example, the dark meats, the legs, the thighs, have more mitochondria than the light meat or the breasts do. And if you think about it, the legs and the thighs do more work than the breasts in a chicken. Rigor mortis is actually due to a lack of ATP. ATP is required for muscle contraction and muscle relaxation. So what happens when you die is your muscles all contract, but then you're dead, so you can't make any more ATP. So your muscles can't relax until eventually the ATP, everything in your body, your cells, starts to degenerate and your muscles will relax. But they can actually tell time of death sometimes based on if somebody is in rigor mortis or has already been through rigor mortis, because of the time it takes the muscles to contract, stay contracted, and then relax after everything is broken down. Okay, anaerobic fermentation if we do not have oxygen present. Pyruvate is that pivotal metabolite in respiration. So the driving force affecting that fate is whether oxygen is present or not. So if oxygen is present, the pyruvate goes through the preparatory stage, then the Krebs cycle, then the electron transport chain. However, if oxygen is not present, the pyruvate goes through fermentation, and it can be lactate or alcoholic fermentation. So oxygen is not going to be used, water is not going to be formed. So with fermentation, you still have glycolysis happening, so glucose goes to pyruvate and gives you 2 ATP. But then if oxygen is not present, we will either have alcoholic fermentation or lactic acid fermentation. In yeast cells, they go through what's called alcoholic fermentation. So carbon dioxide plus ethanol. Again, yeast cells can be used to brew beer and bread and all sorts of things. But this is what they do if they do not have oxygen present. If oxygen isn't present in animal cells, like us, we go through what's called lactic acid fermentation. What happens is we get an oxygen debt, and the pH decreases and our muscles get really tired. So lactic acid starts to build up and you feel that burn. So if you're exercising and you're not breathing properly and you feel that burn, that's lactic acid building up. Some of the lactic acid is actually converted back to pyruvate in the liver. Others are not. So if you are exercising and you do start to feel that lactic acid build up, that burn, you need to start breathing properly to get oxygen to your cells or stop exercising. Both products of fermentation are toxic. So that lactic acid is toxic. The ethanol is toxic. So as I said, lactic acid, we can recover somewhat because the liver can convert some of that back to pyruvate. However, if the buildup is too great, you can actually damage your muscles. So again, oxygen recovery, anaerobic cellular respiration. So start breathing properly. Now we have some advantages and some disadvantages of anaerobic fermentation, basically. Anaerobic respiration gives you a rapid burst of energy. It's fast, rapid burst. However, it's short-lived. It doesn't give you a lot of ATP. It's only 2% efficient, and the products are toxic. So sprinters would use anaerobic respiration to sprint that 100-meter dash, or whatever it's called these days. Long-distance runners need to breathe and use aerobic respiration to make it 1,600 meters, or whatever it is. So it's the difference between a sprinter and a long-distance runner. So sprinter, anaerobic, long-distance runner, aerobic. Sprinters, if they run any great distance without breathing, they're going to get that lactic acid buildup. So you need to know for your test, advantages of anaerobic are that rapid burst of energy. Disadvantages are it's short-lived. You do not get a lot of ATP, so it's not very efficient. And the products are toxic. Lactic acid and ethanol are both toxic. Economically speaking, yeast, we can make beer, wine, and bread, as I said earlier. Anaerobic bacteria can make cheese, yogurt, sauerkraut, acetic acid, isopropanol, a lot of things. So economically speaking, we can benefit from anaerobic fermentation. So the sun comes in, photosynthesis occurs, glucose is produced. Glucose will then go through glycolysis to become 2 pyruvate molecules. If oxygen is present, cellular respiration will occur, aerobic cellular respiration will occur. Glucose will be completely oxidized. Water and carbon dioxide will be byproducts, and the net energy is 36 ATP. However, if oxygen is not present, that pyruvate molecule will go through fermentation. Glucose will not be completely oxidized. Waste products will be lactic acid or ethanol and carbon dioxide, which are toxic, and the net energy is only 2 ATP. So there's a big difference between aerobic and anaerobic. So metabolism, remember, is the sum of all chemical reactions in our body. Anabolism is building things up. Catabolism is breaking things down. So when you add your buildups and your breakdowns, you have metabolism. So anabolism is synthetic, meaning you're building things up. Catabolism is degradative, meaning you're breaking things down. So catabolic reactions release energy, which can then drive anabolic reactions, which require energy. Other types of molecules can be used to generate ATP, like proteins and fats. but they're going to enter at different points during the respiration cycle. So proteins will be broken down into amino acids. Lipids and fats will be broken down into fatty acids. The amino acids will then enter the Krebs cycle. The fatty acids will go to acetyl coenzyme A. Pyruvate can be involved, it just really depends. And then the Krebs cycle will continue and make different metabolic products. We have non-essential versus essential amino acids. Non-essential amino acids are amino acids that our body can synthesize. Essential amino acids are amino acids that our body cannot synthesize, so we have to have them in our diet. Eggs, dairy, poultry, and seafood provide all of the 9 essential amino acids, which are isoleucine, leucine, valine, histidine, lysine, methionine, phenylalanine, threonine, and tryptophan. Then the non-essential amino acids our body can synthesize. So prolonged aerobic exercise does burn fat, but it requires oxygen. If the oxygen runs out, the cells will switch to fermentation and the fat burning is going to decrease. So if you want to lose those extra pounds, you have to have aerobic exercise. Brisk walking, bicycling, swimming, jogging, but very importantly, breathing in and out. That is crucial. Other benefits of aerobic exercise: your heart and lungs become stronger, you get more oxygen to your muscles. Your skeletal muscle, the blood supply is going to increase, the number of mitochondria will increase, and muscles can use more energy even when the body is at rest. So exercise is extremely important as long as you breathe. That is the biggest thing.